So my name is James J. Hickman. I'm at the University of Central Florida in the Nanoscience Technology Center. What I'm trying to do is create functional in vitro systems, which is creating something between a single cell and an animal or human. Build these body-on-a-chip-based systems in collaboration with Mike Schuler at Cornell University to really replace animals in the drug discovery and toxicology process. It was a gap there in terms of trying to mimic what was going on in vivo. And so my training as a surface chemistry uniquely made me able to marry up biological and non-biological materials. It has progressed from looking at trying to create neuronal systems for artificial intelligence applications to then adding in muscle for looking at neuromuscular junction, and now we're trying to do 10 organs on a chip. We did a lot of this work with animals. It works very, very well, but there's just gaps out there in terms of the availability of certain cells. And certainly it's very hard to get a hold of primary neurons, and primary cardiomyocytes. And so the induced pluripotent stem cells allows you to bypass using a lot of primaries, but also eventually what we want to do is look at diseases, right? And so the induced pluripotent stem cells have the advantage of, if you can take it from a diseased patient, you might be able to reproduce the disease state, and then you'd be able to have a control based upon something that we've built as a functional in vitro system, and then be able to have the disease cells to be able to compare it to, and then use that directly as a phenotypic assay to test drugs. The only way to address this right now is through whole animal work or testing on human beings. And so since we're very limited in what kind of testing we can do on human beings, there's not many other ways to approach this other than trying to build this out of human cells. The human cardiomyocytes have allowed us to have another cell in our arsenal. We also work with the iCell neurons, and that's been the only source of neurons we've been able to have. By having both of those, we now have a pretty defined source of both cardiomyocytes and neurons to put into our systems. And again, the hope is, as CDI starts going forward, is to have the diseased models for those cells as well. And that'll be really interesting to be able to start testing the controls versus the diseased ones. The technical support folks at CDI have been great. They're very responsive. They're always there to try and help. We've had a good working relationship with them. We do webinars, they go back and forth, and they get excited about what we're doing as well, and that's always so good. And by having access to the diseased states using these cardiomyocytes, that's going to be a big area. And also the genetic diversity, male versus female, age effects from these cells will be very, very important. So by having it as a pre-screen and being able to have available the wide variety of genetic diversity as well as the disease states will be allow it to get a lot more well incorporated into the drug discovery, toxicology, and just basic research.